15.3, we're going to be looking at linked genes, and linked genes implies that two genes are tied together, thus they are inherited together. So we're going to be looking at linked genes, and what causes them to be inherited together is they are found on the same chromosome, and they are very, very close together on that chromosome. So once again referencing traits that are inherited as a combo, and they are found on the same chromosome. Recall that the X chromosome has about 1,100 genes, and the Y chromosome has only 78 genes. Autosomal chromosomes, the other 22 pairs of chromosomes, each contain hundreds of thousands of genes. So those do operate considerably differently than the sex-linked genes we were looking at in 15.2. So we're going to look at how linkage affects inheritance by revisiting Morgan and his experiment, talk a little bit more about what he did. He looked further with flies to see how inheritance of two different characters could be linked together. So to do this, Morgan looked at two characters. He looked at body color and wing size. Each of these has two different phenotypes. The wild type flies that we see right here have gray bodies and normal size wings. And when I say wild type, I'm simply talking about the standard usual phenotype. So what you typically would see in these type of flies. These flies happen to be true breeding, which meant that they would mate with another fly of the same genotype and the same phenotype. What he did with these true breeding gray normal winged flies is he crossed them with doubly mutant flies. He obtained these doubly mutant flies from previous controlled breedings to get this particular desired outcome that he wanted, presumably for this specific cross. So looking at their genotypes, we can see that this fly is homozygous for a gray body color and homozygous for normal wings, whereas a doubly mutant is homozygous for a black body and homozygous for vestigial wings. Vestigial wings are more or less wings that are present but have zero functionality because they're so shriveled up. So that's a vestigial wing if you were wondering that. As a result of crossing these two flies in this P generation, he resulted in an F1 cross, and each of the flies had the standard wild type phenotype, but they were heterozygous for each of the traits, the body color and the wing color. What he then did is took the F1 phenotype and once again crossed it again with another double mutant fly. So homozygous for black, homozygous for vestigial wings. So keep in mind when we're creating gametes for this fly and gametes for this fly, these genes can arrange themselves in one of four ways, and really these genes can only arrange themselves in one way. So let's take a look at the different combinations that these alleles could arrange themselves. So if a fly happens to be B plus B and VG plus VG, the possible gametes could be B plus VG plus, B plus VG, B VG plus, or B VG. And we see those right down here. And this would be for the F1 flies. And the double mutant flies, since all the alleles would be the same, regardless here or here, all the gametes would be little b VG. So if we were to take those possible gametes and plug them into a dihybrid cross here, you can maybe visualize that since that is, well, B plus, VG plus, and all these are the same, all four of those boxes would have the same possible recombinant cross. And same thing with this, all four of these boxes would have the same thing. And same with these ones, and same with these ones. So it would be a safe assumption that if we were to actually mate these flies, we would see 4 to 4 to 4 to 4 ratio, or if we were to reduce that, it'd be a 1 to 1 to 1 to 1 ratio. Now, in order for us to get that particular ratio, these two alleles would have to assort independently from one another. And keep in mind that in order for genes to assort independently, they must be on different chromosomes. But what would we expect if the alleles happen to be on the same chromosome and are inherited together? How would that affect our expected ratio? So going back to this image, we can see that the F1 is a blend of both parents in the P generation. And we see that blend right here. But what if the genes for the Bs and the genes for the VGs are on the same chromosome? And better yet, what if they were on the same chromosome and located close together? Well, that would mean that the F1 would have this B plus and this VG plus linked on the same chromosome because they came from this fly, and the B and the VG would be on the second homologous chromosome because they came from this fly. Now once again, keep in mind that we're talking alleles here, and if this allele comes from one parent, then this matching allele will come from the second parent, and they're on homologous chromosomes. So they're on actual different chromosomes. We just write them this way because they are for the same gene. They're found on the same chromosome at the same locus. So when we see this right here, we are looking more at something kind of like this image right here, where we can get a little bit better picture of that. The B plus and the VG plus are on the same chromosome, and the B, VG, they're on the same chromosome. And this would have come from the wild type fly. This would have come from the double mutant fly. So we do see that this fly would be heterozygous for the Bs as well as heterozygous for the VGs, which is actually what we're seeing written right here. So if these were in fact found on the same chromosomes like this, and these two alleles did not separate independently from one another, if they were kind of stuck together, how would that affect our expected outcomes? Well, let's go back a few slides here and take a look at these gametes. Keep in mind that the B plus and the VG are linked together, and the B and the VG are linked together. What that would mean is that these possible gametes wouldn't exist. We would have two of them, but not the other two. So we could essentially go through and delete this one because this B plus could not link up with that VG because the B plus is linked to the VG plus. So they're on the same chromosome, so this one could not meet up with this one. Well, that would also mean that the little b here cannot link up with the VG plus, so we can get rid of that. And that would mean that when we're going through gamete formation, the fly can either pass on the B plus VG plus or the B VG because, going back a few slides, they are linked together. And that's what this image right here shows us. This, in this case, female fly who's heterozygous can either pass on this red chromosome and pass on those two genes linked together, or pass on this whitish pink chromosome with those two alleles linked together. So taking a look at our cross, if those two alleles are linked together right here, and if those two alleles are linked together right here, what does that do to our expected outcome? Well, would we have any flies that are B plus VG? We wouldn't expect any because those two alleles are found on different chromosomes. They're not linked together. What about this combination? Well, that gamete wouldn't exist either because that little b and the VG plus, they're on separate chromosomes as well. So the fly from the F1 could either pass on this particular chromosome or that particular chromosome, and the doubly mutant fly would of course pass on the same gamete. So what we would expect is we would expect 50% of the offspring to be B plus B, VG plus VG, and the other 50% to be B, B, VG, VG. We wouldn't expect any to have the B plus B, VG, VG phenotype, or genotype I should say, and we would expect none to be little b, VG plus VG, because those two gametes shouldn't exist if we have linkage going on. So we would have essentially a 1 to 0 to 0 to 1 ratio. And another way to think about this is that all of the F2 would have the parents' genotype that were used from the F1 cross. Keep in mind that the normal wild type fly, it was a heterozygous for the Bs, it was heterozygous for the VGs, and the second parent, the doubly mutant one, was homozygous for the little b, and homozygous for VG. Let's take another look at those parents right here. We can see that homozygous right there and right there. Heterozygous, sorry. And we can see the homozygous right here and right here. So what did Morgan actually get from his results? Well, he was expecting 50% to be the gray wild type heterozygous genotype, and he was expecting the other 50% to be the homozygous doubly mutant wild type, or the parental genotypes is what he was expecting. What he actually got, though, was a few that had the recombinant genotype, so B plus B, VG, and the BB, VG plus VG, which we said right here shouldn't exist because of the linkage. Well, his actual numbers kind of hinted at, well, they are linked. So, in essence, he was right. What he got was 965 flies that had this parent's genotype. He got 944 flies that had that parent's genotype. And he got a few flies that were the recombinant genotypes. 206 of these ones, 100 looks like 85 of these ones. So he did get a few of these flies gametes produced, and he did get a few of these flies gametes produced, which is kind of odd. He wasn't expecting that. So with the actual data, 965, 944, 206, 185, what does that data suggest? Well, to start with, is the data anywhere close to the expected 1 to 1 to 1 to 1 ratio that we would see if the genes were not linked? No, it's not even close. These numbers are way different from one another. But are they pretty close to the 1 to 1 to 0 to 0 ratio? Well, they're not really close, but they're shockingly close. I don't know if that makes sense. They're pretty close to suggest that there is some linkage going on. And that's exactly what he kind of concluded. He concluded that these numbers are so much higher to the parental genotypes because these alleles are linked together on the same chromosome, and these alleles are linked together on the same chromosome, which we saw in this illustration right here. We saw a little bit of linkage. So this image right here just kind of shows us the wild type fly mated with that mutant fly, and we can see that all the children expected, if there is linkage, would have this red chromosome mixed with a blue one, or would have this pinkish white chromosome mixed with a blue one, which is why we saw most of the flies having this particular genotype phenotype or this genotype phenotype.